Hello everyone, welcome to the Swayam course of Genetic Engineering and its recent advancement. I am Dr. Richa Sahu and I am working as an assistant professor in MIT Institute of Biotechnology, MIT University, Chhattisgarh. So today we will be talking about basic instrumentation in genetic engineering. So till now we have talked about various concepts of genetic engineering, what is the recent technologies, what is the basic concept of genetic engineering, everything we have covered. So today we will be talking about the instrumentation part. So genetic engineering is a field of biotechnology that involves the manipulation of an organism's genetic material to achieve specific outcomes such as creation of genetically modified organisms, also known as GMOs, the development of biopharmaceuticals and the study of gene function. To carry out these experiments or tasks, rather I should say, effectively, scientists and researchers and the workers rely on a range of specialized instruments and equipment. These equipments and instruments allow them to isolate, analyze, modify and manipulate DNA and other genetic materials. Here I will be talking about some key and basic instruments that are used in genetic engineering and its experiments. The first instrument or the technique that we will be talking about is PCR machine or thermal cycler. So, polymerase chain reaction or PCR is a fundamental technique in genetic engineering, and the PCR machine or thermal cycler is used to automate the process of DNA amplification. It rapidly heats and cools DNA samples to replicate specific DNA sequences, allowing researchers and the workers and the people who are working on it to create millions of copies of target gene. Thermal cyclers are known as PCR machines, are the essential instruments in genetic engineering and molecular biology for performing the polymerase chain reaction, also known as PCR. So, PCR is basically a technique used to amplify specific DNA sequence. So, what are the things that are required for a PCR to carry out? It requires a DNA that has to be amplified, it requires a primers, it requires DNTPs and an enzyme DNA polymerase or Taq DNA polymerase. The thermal cycler automates the temperature cycling required for PCR. Here I will explain the principles and working of a thermal cycler in detail. Starting with the principle on which the thermal cycler or the PCR machine works. The PCR machine or the thermal cycler basically operates on the principle of thermocycling, which involves repeatedly changing the temperature of the reaction mixture to facilitate DNA denaturation, primer annealing and DNA extension. These are the three basic steps that brings about the PCR or the polymerase chain reaction. These temperature changes are critical for amplifying a specific DNA region using a heat stable DNA polymerase enzyme Taq polymerase. Talking about the working of the PCR machine or the thermal cycler, a typical thermal cycler goes through a series of temperature steps, typically including three basic steps starting with denaturation, annealing and then extension, repeated for a set of number of cycles. Here is how it works, starting with the first step, that is initialization. In initialization, the thermal cycler or the PCR machine initially heats the reaction chamber to a high temperature, typically around 95 degrees centigrade. This step helps to ensure that any remaining double stranded DNA in the sample is fully denatured, providing single stranded templates for PCR. So, the single stranded DNA that has been formed in this particular step will be acting as a template for the polymerase chain reaction. The next step is denaturation, also known as melting. The temperature is raised to around 94 degrees centigrade to 98 degrees centigrade for a short interval of time, like usually 20 to 30 seconds. At this high temperature, the hydrogen bonds holding the two DNA strands together break, causing the DNA to denature into single strand, followed by the third step, that is annealing. the temperature is lowered to around 50 to 65 degrees centigrade depending on the primer sequencer that has been taken or used for 20 to 30 seconds. This allows the DNA primer to bind or to anneal to their complementary sequences on the single stranded DNA template. Followed by the next step, that is extension, also known as elongation. Here the temperature is raised to approximately 72 degrees centigrade for 20 to 30 seconds. At this temperature, a heat stable DNA polymerase enzyme, namely Taq polymerase, adds complementary nucleotides to 3' end of the primers, synthesizing a new DNA strand, followed by the next step, that is cycling. Step 2 to 4 are repeated for a specified number of cycles, typically 20 to 40 cycles depending on the amount of DNA that is required for the experiment to carry on. So, after 20 to 40 cycles around, to exponentially amplify the target DNA region. Each cycle doubles the amount of DNA in that reaction. And finally, final extension, the last step. After the cycling is complete, a final extension step is often performed at 72 degrees centigrade for a few minutes to ensure that all partially synthesized DNA strands are fully extended. Finally, cooling. The thermal cycler cools down to around 4 to 15 degrees centigrade to hold the reaction at low temperature, preventing DNA re-annealing. The thermal cycler's precise control of temperature and time for each step is a crucial for success of the polymerase chain reaction. This automation allows for the rapid and highly reproducible amplification of specific DNA sequences, making it a fundamental technique in genetic engineering, diagnostics and various molecular biology applications including DNA sequencing, gene expression analysis and DNA cloning. The next basic instrument that we will be talking about is a centrifuge. So, basically centrifuge are the instruments that are used to separate different components of a mixture based on their density. So, centrifuges basically work on the principle of sedimentation, and under the influence of centripetal acceleration, the components of a mixture are separated on the basis of their density. In genetic engineering, centrifuges are crucial for isolating DNA, RNA and other proteins from cells, tissues. High speed centrifuges are used for pelleting cellular components, while ultra centrifuges are used for more precise separations. So, depending on the sample that we have, we choose the type of centrifuge to be used, and also they have different rotors having different speeds and different volumes of sample that they can hold. The next instrument that we will be talking about is electrophoresis system. So, basically electrophoresis is a technique which is used to separate DNA, RNA or proteins on the basis of their size and charge. We will be talking more about this in detail. An electrophoresis system typically includes an agarose or polyacrylamide gel and an electrical or electric field. DNA fragments migrate through the gel, allowing researchers to analyze and purify DNA samples. So, to talk more about it in detail, so basically gel electrophoresis is a widely used technique in molecular biology and genetics for the separation and analysis of macromolecules, primarily DNA, RNA and proteins. It operates on the principle of charge and size. That means, on the basis of different charge and size of the molecules, they are separated, allowing molecules to be separated based on their mobility in the influence of electric field through a gel matrix. Here I will explain the principle and working of a gel electrophoresis in detail. So, as I have mentioned before, gel electrophoresis relies on the principle of the fact that charged molecules such as nucleic acid, DNA or RNA and proteins migrate in an electrical or electric field rather I should say. The rate of migration of these macromolecules or these molecules depends on the charge, size and shape of the molecules. A gel matrix usually made up of agarose or polyacrylamide is used to impede the movement of these molecules through the porous structure that they have. Smaller the molecules, they move faster on the gel or more quickly than the larger ones, resulting in separation based on the size. Talking about working of gel electrophoresis, the gel electrophoresis process involves several key steps. The first step is the preparation of gel, that may be agarose or polyacrylamide. A gel is prepared by mixing powdered agarose or acrylamide with a buffer solution. In acrylamide we use two things, bisacrylamide and acrylamide in a buffer solution if you are working with page. So, there are two types of electrophoresis, age and page. Age is agarose gel electrophoresis, page is polyacrylamide gel electrophoresis. So, now the gel mixture is heated and poured into casting tray with comb like structures to create wells for the sample loading. After the gel solidifies, it forms a porous matrix through which the biomolecules can migrate under the influence of electric field. The next step is sample loading. So, for that you need to prepare the sample like DNA, RNA, whatever you have taken or proteins. The DNA, RNA or protein samples are mixed with a loading buffer which typically contains a tracking dye for visualization and a denser substance like glycerol to make the samples heavy a little bit so that they can sink into the well, so that they should not come out when you put the samples into the well. The samples are loaded into the wells at one end of the gel. Now, the setup of the electrophoresis unit. The gel is placed in an electrophoresis chamber filled with an electrically conductive buffer solution. The electrode, usually made up of metal, are immersed at both ends of the chamber and electric current is applied. The negatively charged biomolecules move towards the positive electrode, that is anode, due to the electric field. As the current flows through the gel, the biomolecules migrate through the gel matrix at rates determined by the size and charge. Smaller molecules move more quickly and travel farther through the gel, while larger molecules move more slowly and remain closer to the starting point. After the gel has been run on the electrophoresis unit, the next step is visualization. How do you visualize it? After the electrophoresis, the gel is removed from the chamber and stained with a dye. The dye can be ethidium bromide for DNA or labeled probe like or antibodies for proteins. There are different types of stain that are used. There can be a silver stain or a blue stain. The stained or the labeled molecules are then visualized under UV light or by other means depending on the type of biomolecule that you are analyzing. The resulting pattern of the separated molecules appears as bands or lanes on the gel. By comparing the migration distances of sample bands to those of known molecular weight markers or standards, researchers or the people working on it can estimate the size of the or the molecular weight of the biomolecules. Gel electrophoresis in genetic engineering is used for a wide range of applications, including DNA fragment analysis like DNA fingerprinting, restriction enzyme digestion, RNA size determination, protein separation to assess the purity of the protein. It is the fundamental tool in molecular biology and genetics laboratory for characterization and quantifying the biomolecules. Moving ahead with the next instrument. So, the next instrument that we will be talking about is a spectrophotometer. So, spectrophotometers are used to measure the absorbance of light by a sample. In genetic engineering, they are employed to determine the concentration of nucleic acid, DNA or RNA and proteins in a solution. The information is very crucial for various experimental procedures. Spectrophotometers are essential instruments in genetic engineering and molecular biology for quantifying the concentration of biomolecules, including DNA, RNA and proteins. They operate on the principle of measuring the absorbance of light by biomolecules or molecules at a specific wavelength. So, here I will be talking about the detailed explanation of the principle and working of a spectrophotometer. So, basically talking about the principle of spectrophotometry, it is based on the Beer-Lambert law, which states that the absorbance of a solution is directly proportional to the concentration of absorbing species or the biomolecules and the path length through which the light passes, and inversely proportional to the intensity of the incident light. So, we can write it as A is equals to eta Cl. A is the absorbance, epsilon is the molar absorptivity or molar extension coefficient, a constant that describes how strongly a substance absorbs light at a particular wavelength. Small c is the concentration of the substance in the solution. L or small l is the path length of the light through the sample. Capital I is the intensity of the incident light. So, this was the principle of the spectrophotometer. Talking about the working, the operation of a spectrophotometer involves several key steps, or there are few important parts of the spectrophotometer I should first discuss. The first is the light source. Spectrophotometers typically have a light source that emits a broad spectrum of light, often in the range of UV and visible. Some instruments cover both UV and visible region, and some instrument or some spectrophotometer only cover the visible region or sometimes the UV regions. The next thing is monochromator. The emitted light from the light source is passed through the monochromator, which allows only a narrow range of wavelength to pass through. This is important for selecting the specific wavelength of light that matches the absorbance peak of the molecule being analyzed. The next thing is the sample and reference cells or the blank cells. These are the cells where you put the samples in the cuvette. So, basically spectrophotometers use two cuvettes or cells, a sample cell and a reference cell, also known as a blank cell. The sample cells holds the solution to be analyzed, while the reference cells contain a solvent or a buffer without the analyte, basically to take it as a blank. The reference cell compensates for variations in the intensity of the light source. Next is the absorbance measurement. Light passes through both the reference and sample cells. The spectrophotometer measures the intensity of the light before and after passing through the sample. The difference in the intensity between the two measurements is proportional to the absorbance of the sample. Next is a very important thing that has to be done is calibration. Before analysis, the spectrophotometer is often calibrated using a blank solution. That means, the reference cell filled with the solvent only to zero the instrument and to ensure the accurate measurement. The instrument calculates and displays the absorbance of the sample at the selected wavelength. The concentration of the analyte can be determined using the Beer-Lambert's law if the molar absorbability, if the molar extension coefficient is known. You can also plot a standard curve for a particular analyte, so that on the basis of absorbance we can take out or we can calculate the concentration of the analyte. Next is the wavelength selection. Spectrophotometers allow users to select the desired wavelength of light to match the absorbance maxima of the analyte, which is known from its spectral characteristics. So, what are the applications of the spectrophotometer in genetic engineering? So, spectrophotometers are widely used in genetic engineering for various applications, like for example, quantification of nucleic acid, Spectrophotometers measure the absorbance of DNA or RNA at 260 nanometer to determine the concentration and purity. Similarly, it is also used for the protein quantification. Protein concentration can be determined using spectrophotometric methods, often measuring absorbance at 280 nanometer if you are not using any color producing dyes or color producing chemicals along with the proteins like phosphoenolaurin. In that we measure the absorbance at a visible range. Otherwise, for protein measurement the wavelength that is to be taken is 280 nanometer. Next is enzyme kinetics, very important application. Spectrophotometers are used to monitor enzyme catalyzed reaction by measuring changes in the absorbance over time. Next is cell culture. Absorbance reading can be used to estimate cell density in microbial cultures. There are many other applications, these are the major application that has been mentioned here. Spectrophotometry is basically a fundamental tool in genetic research, allowing precise and rapid quantification of biomolecules that are essential for experimental design and analysis. The next instrument or next technique that we are talking about is DNA sequencer. DNA sequencing instruments are used to determine the order of nucleotide bases in a DNA molecule to be very precise and simple. Modern DNA sequencers employ advanced technique such as next generation sequencing or NGS to rapidly and accurately determine the sequence of the entire genome. DNA sequencers are the instruments used to determine the precise order of the nucleotide bases adenine, cytosine, guanine, thymine in a DNA molecule, which is known as DNA sequencing. DNA sequencing is a fundamental technique in genetics, genomics and molecular biology. Now, I will explain the principle and working of a DNA sequencer in detail, starting with the principle on which the DNA sequencer works. The DNA sequencer operates on the basis of several different sequencing methods, but one common principle underlying all of them is the generation of a series of nucleotide specific signals. For example, a fluorescent signal that can be read and translated into the DNA sequence. The specific method used can vary, with Sanger sequencing and next generation sequencing being the two primary approaches. Talking about the working of a DNA sequencer, if it is based on Sanger sequencing. So, Sanger sequencing, also known as chain termination sequencing, is one of the traditional methods for DNA sequence. The first step is the sample preparation. The DNA sample to be sequenced is purified and denatured into single strand. The next step is primer annealing. A DNA primer with a known sequence is annealed or bound to the single stranded DNA template. This primer serves as the starting point for DNA synthesis. The next step is DNA synthesis. The enzyme used is DNA polymerase. The DNA building blocks or nucleotides and a mixture of chain terminating dideoxynucleotides or DDNTPs are added to the reaction. Regular nucleotides or DNTPs incorporate into the growing DNA chain, but DDNTPs lack 3' hydroxyl group needed for further chain extension, causing DNA synthesis to terminate randomly at positions corresponding to the incorporated DDNTPs. Next is the fragment separation. The reaction results in the mixture of DNA fragments, each terminating at a different positions. These fragments are separated by size using gel electrophoresis, creating a series of bands on the gel. The next step is signal detection. The gel is exposed to a laser or a UV light source, causing the DNA fragments to fluoresce. If they are labeled with fluorescent tags or any other tags depending on the type of tagging that are used in DDNTPs. The fluorescence emission is detected and recorded as peaks in the chromatogram, with each peak representing a specific base, a specific DDNTP. Next is the base calling. The software analyzes the chromatogram to determine the sequence by interpreting the order of the peaks. Each peak corresponds to one of the 4 DNA bases, that is adenine, cytosine, guanine and thymine. Talking about the working of the next generation sequencing or NGS. NGS methods such as Illumina sequencing differ significantly with Sanger sequencing. These methods involve massively parallel sequencing where millions of DNA fragments are sequenced simultaneously. First is the library preparation. DNA samples fragmented into shorter pieces or shorter fragments and adapters are ligated to the ends of these fragments. So, these shorter fragments that are generated, the adapters are ligated to these shorter fragments. Next is cluster generation. The DNA fragments are immobilized on solid surface such as flow cell. Each fragment forms a cluster of identical DNA molecule. Now, sequencing by synthesis. A series of 4 different fluorescently labeled nucleotides are sequentially added to the cluster. As each nucleotide is incorporated into the growing DNA strand, it emits a signal that is detected by the camera. Finally, the base calling. The sequence of each cluster is determined based on the order of nucleotide incorporations and the corresponding fluorescence signals are recorded. The data from all clusters are combined and processed to generate a DNA sequence. Next generation sequencing methods or NGS methods allow for high throughput sequencing, making it faster and more cost effective for whole genome sequencing, metagenomics and various other applications. DNA sequencer are the pivotal tool in genetic research, enabling scientists to decode and the genetic information of the organisms and study genomes, mutation and genetic variations. The next instrumentation that we will be talking about is electroporator. So, basically an electroporator is a laboratory instrument used in molecular biology and genetic engineering to introduce DNA, RNA or other macromolecules into cells by temporarily increasing the permeability of the cell membrane using electrical pulses. This process is known as electroporation. Here is the detailed explanation of the principle and working of the electroporator. So, basically electroporator is based on the principle of application of brief high voltage electrical pulses or electric pulses to cells which create transient pores or channels into the cell membrane. These pores allow molecules such as DNA or RNA to pass into the cell. Once the electric field is removed, the membrane reseals, trapping the introduced biomolecules inside the cells. Electroporation can be used to introduce genetic materials, drugs or other molecules into a wide range of cell types. Talking about the working of an electroporator, the first step is preparation of the cells. The cells to be electroporated are prepared and harvested in a suspension or as a monolayer depending on the cell type and application. Next is cell suspension formation and molecular and molecule mixing. The cells are mixed with the molecule of interest like DNA, RNA or drug that you have taken in an electroporation buffer. The buffer helps to maintain cells viability during the procedure and ensures uniform electrical conductivity. For electroporation setup, the cell molecule mixture is placed in specialized electroporation cuvette or chamber with parallel electrodes. The cuvette is carefully positioned in the electroporator to ensure the cells are evenly exposed to the electric field. So, how do you set the pulse parameters? The user sets the electroporation parameters including the voltage, pulse duration or time and the number of pulses. These parameters vary depending on the cell type, molecule and the electroporator model. When the electroporation is initiated, a high voltage electrical pulse is applied across the electrode. The electric field disrupts the lipid bilayer of the cell membrane, creating a transient pores. Why transient? Because when the electric field is withdrawn, they reseal themselves. This pore allows the molecule from the surrounding medium, the electroporation buffer that is, to enter the cell. In the case of genetic engineering, this is where the DNA or RNA molecule are introduced in those cells. For the recovery of the cells after electroporation, the cells are immediately transferred to a recovery medium, that is the growth medium or culture medium, to allow the cells to heal and membrane to reseal. Now, finally, the cell culture and the analysis. The electroporated cells are typically cultured for a period to allow the introduced molecules to be expressed or incorporated into the cell's genetic material. Researchers can then analyze the cells to study the effects of introduced molecules such as gene expression or functional changes. The success of electroporation depends on optimizing parameters such as voltage, pulse duration and buffer composition for the specific cell type and application. Electroporation is widely used in various fields including molecular biology, biotechnology and gene therapy to manipulate and study cells for research or therapeutic purpose. So, the techniques that we talked about today are the basic techniques and the basic instruments that are used in genetic engineering in day-to-day laboratory activity. So, I hope you understood the basic principle and working of these techniques. Thank you very much. We will meet again in the next lecture.